We are now recording, and thank you very much, Ben, for agreeing to give about the cell seminar, and it's all yours.
Sure. Well, thanks for giving me the opportunity to give this talk. I'm excited because I was looking through the audience, and I don't think I recognize hardly anyone, maybe a few people. So it's exciting to have the opportunity to speak to a new audience. But I'm Ben Larson. I'm a postdoc in Wallace Marshall's lab at UCSF. I did my undergrad in physics at Reed College in Portland, Oregon, and started sort of making my transition into biology. I worked at the National Institutes of Health in the lab of Justin Tarasca. And I did a lot of microscopy and computational image analysis there, looking at endocytosis and exocytosis. And then eventually went on to UC Berkeley to do my PhD with Nicole King, where I really immersed myself in biology. And I worked on the question of how animals evolved from unicellular ancestors by studying these critters called 𝗂𝗏𝗵𝘃𝗹𝗏𝗂𝗏. They're the closest living relatives of all animals. And it was during that time that I got the opportunity to do field work. I feel really lucky that Nicole let me get out there in the world and look for cells in the environment. And that has started this really fascination for me that I guess my former and current lab mates might call more of an obsession with protists, microbial eukaryotes, and all the amazing things that they do. And so today I'm going to be telling you a bit about the interesting behavior of one particular kind of protist called Euplotes that you can see here in this banner crawling around. Okay. Oops. So just to set the stage a little bit, I probably don't need to tell this audience, but life is fundamentally cellular, and across any scale of space and time that you want to pick, pretty much any biological process or biological event can be meaningfully reduced to things that cells are doing. And it's easy to get the idea that the main way that cells do complicated and interesting things is by getting a bunch of cells together to achieve some more complex biological process. Of course, animal development is incredible, and brains are pretty cool as well. But in fact, even single-celled organisms can display remarkably complex and sophisticated, even animal-like behavior. So what you're watching here is actually a single cell on the right here. It's a ciliate that's devouring its animal prey. And this is a scene that's not so hard to find if you were to just sort of look in pond water. And so, I think there's this real challenge that we're faced with when we're trying to understand something like complex cell behavior, maybe like a crawling neutrophil-like cell over here, where we're gaining an increasingly complex and sophisticated understanding of all sorts of molecular details underlying cellular processes. And we know that cellular behavior emerges from these kinds of molecular processes in conjunction with interactions with the cellular environment. But there's great difficulty still in sort of making sense of all of these complex details. So how do we understand something like cellular behavior without getting lost in all of these kinds of details? And the situation might look almost worse if we consider the diversity of cells that we might find. So here is just a bunch of snapshots of cells that I found in various field samples. And while this complexity and diversity presents a challenge, I think it also presents an opportunity. And something that Wallace Marshall's lab is really great at is finding just the right kind of often strange cell to highlight particular kinds of biological questions of interest. And so the perhaps somewhat strange cell that I've chosen to focus on for a lot of my postdoc work is this organism called Euplotes that I mentioned. So you can see here the cell, you're sort of looking at the interface of the cell in a cover slip. And you can see that it's moving these leg-like appendages to actually walk across a surface. And so, of course, it presents this interesting question of how can a single cell coordinate something like this walking behavior. And Euplotes, while it is a complicated cell, you might see all these asymmetries and high complexity and structure in the cell. It actually is quite amenable to quantitative analysis. For one thing, a lot of the things that it does, these things happen at interfaces. So like I can watch it relatively easily as it walks across a cover slip, for example. And it also has a highly stereotyped structure, cellular structure. So I can find positions on the cell and the orientation of the cell from organism to organism. So this sort of discrete and highly stereotyped cell structure makes it very amenable to quantitative analyses. And as you can see, I'd love to understand how it does all sorts of these complicated maneuvers, reorienting itself to land on a surface. I'm focusing for now just on its walking behavior, but in the future, it'd be great to understand more about the complex behavioral repertoire that it has. And most of its movements are achieved through the action of these leg-like appendages that I mentioned. They're called cirri. And I mentioned that this cell is a ciliate, and you might wonder, it doesn't look very hairy, where are all the cilia? Well, it turns out these cirri, if you take a cross section through a cirrus, sort of just below the cell cortex, this is the kind of structure that you'll see, and it's this highly ordered arrangement of cilia. And so each one of these things is a cilium, and they're linked together by fibers that connect the cilia, and also there are these internal fibers within the cell that connect these cirri. And so these fibers that connect the cirri are actually visible just by light microscopy. And the behavior of these cells was so complex that early on in protostology, researchers thought that they might have some sort of rudimentary nervous system that they called the neuromotor apparatus in order to coordinate the movements of the cell. And with more modern microscopy techniques, this is just tubulin stained here, we can see that they're actually, these fibers are microtubule-based structures in the cell. And one other thing that we know about the movements of Euplotes, you can see here, these are just cell trajectories. There are these characteristic velocity fluctuations, trajectories marked out here. There are these cellular depolarizations that are associated with the movement of the cirri, sort of action potential-like. And the frequency and the amplitude of these depolarizations actually determine something about the movement of these leg-like appendages. Okay, but relatively little is actually known about the details of the movement. And so what I first set out to do is to quantitatively characterize the movement of the cirri during cell walking. And to do this, I started by encoding the sort of activity of cirri at every point in time of walking trajectories of a cell. And as I said before, all of the cirri are distinguishable. So from cell to cell, I can always label them with identifiable labels, label the cirri, that is. And at each point in time, I can just give this 14-dimensional binary vector an entry of a 0 if the cirrus is not moving, or a 1 if the cirrus is flailing around. And then at each time point in my movie, I can go and encode the behavior of the cell like this as a series of these 14-bit binary vectors. And as I said, I'm focusing just on this walking behavior, and you can see this is just a trajectory that a cell is tracing out as it walks, relatively linear. Okay, so how might this thing move? You might expect that it will, even though it's a cell, maybe it'll be somewhat animal-like. And here, this is a fly walking across a cover slip, and this is a representation of its gait. So what the legs are doing, and black corresponds to the leg being lifted off of a surface, white in contact with the surface. And you can see these very highly stereotyped patterns of movement, and it can execute sort of different kinds of gaits, but it's very stereotyped, reproducible, determinate. And you might think, okay, it's a cell, so why should we expect it to walk like an animal? It doesn't have a brain, doesn't have a nervous system, but many unicellular organisms actually execute these kinds of determinate gaits. And this is some nice work from Kirsty Wan, showing all these different flagellates and the different kinds of gaits that they can execute, which are actually somewhat reminiscent of different kinds of animal gaits. But if we actually look at a trajectory of a walking cell, so these are the cirri. I apologize, I labeled them with letters in the other slides, but here they're numbers, the 14 different cirri. Black represents a 0 in that vector that I described before, so the cirrus is not moving, and white represents a moving cirrus. You can see the dynamics are really complicated. And so it's more complicated in this nice determinate case. But if you kind of squint at this, maybe you'll think you can see some sorts of patterns. So how can we get at whether or not there are really patterns in the gait? Well, we can look at a bunch of different cells. So this represents walking trajectories from 13 different cells and many, many time points. And we can do dimensionality reduction. And in this case, I've done non-negative matrix factorization. It's somewhat similar in spirit to PCA, but I can represent in this reduced sort of state space the activity of the cirri over a bunch of different walking trajectories. And what we can see is there's clear structure in the state space. And one advantage to using something like non-negative matrix factorization is these axes, these components are interpretable in that they correspond to particular groups of cirri being active together. And so, for example, H1, this axis here, corresponds to these first three cirri. And this sort of grouping and the dimensionality reduction is in part facilitated by the fact that there's shared mutual information between groups of cirri. So this is just a plot of mutual information. And you can see that some movements of some cirri is highly correlated with one another. And others have weaker correlations, and sometimes there's very little mutual information shared at all between cirri. And so we can perform clustering to capture the structure that we see. There are these groups that we see in the reduced state space, and we find that the clusters that we pick out correspond to particular patterns of cirral activity. So they're unique patterns of cirral activity. And I should also mention here that this clustering, finding optimal clusters is a difficult kind of problem. I can talk in more detail about how we did that if people are curious, but the important thing is that it doesn't matter exactly how many clusters I pick out in this reduced state space. The conclusions that follow are more or less the same. But for what it's worth, in the optimization that I've done, I find 32 gait states. So these are these clusters that represent similar kinds of patterns of cirral activity. Okay, so once we have those clusters, we can then go and look at the dynamics of the transitions between those gait states and ask what that looks like. And I know this is a bit of an intimidating figure. There's a lot that's going on here, but the one takeaway is just it's very complicated. So we have these 32 different gait states, and each node in this graph representation is just the node is scaled by the amount of time that the cell tends to spend in that gait state. The arrows are these edges between the nodes represent the transition frequencies associated with the transitions between any pair of gait states. And these colors of the edges represent the amount of cell movement associated with these transitions. So the cooler colors are less cell movement, hotter colors are more cell movement. And while this graph might be a bit complicated and intimidating, we can break it apart, inspired in part by ideas from non-equilibrium physics. We can look at, based on this idea of what's called detailed balance. So it's a condition of equilibrium. And under detailed balance, transitions in one direction between states will be balanced by the reverse transitions. And so we can break this transition picture of the gaits into unbalanced and balanced transitions. And what we see is that these unbalanced transitions, so this is what's called broken detailed balance, where transitions are much more frequent in one direction versus the other, there are relatively few of these non-equilibrium like transitions given the entire spectrum of possible transitions in the cell. On the other hand, these balanced transitions are super complicated. So these are more like equilibrium fluctuations. And it turns out that most of the cell movement happens during these kinds of balanced transitions. Okay, but the important thing is these unbalanced transitions introduce some sort of a temporal irreversibility. So there's a sort of direction to what the cell is doing. And these are, in fact, the gait states that are involved in these unbalanced type transitions. And we propose that these unbalanced transitions are really what the cell is likely controlling. There has to be some sort of active control in order for the cell to bias its transitions in one direction versus another. Okay. And I guess you can just also see in this plot that some of these states involve cirral activity that's sort of all throughout the cell. Some of these states involve only a few cirri active. But we think these ones are the controlled ones. Okay, so it's kind of hard to wrap your head around exactly what this means for what the gait of the cell looks like. And I'll give one more piece of information, which is this gait state 1 is somewhat special in that it represents no cirral movement. It's almost like a resting state or a reset state for the cell. It's the most dynamically predictable. So we look at the autocorrelation of a binarized signal of this state. And what we find is there's some sort of clear autocorrelation signal, whereas none of the other states have this. So there's some sort of memory that the cell has of this gait state 1. And so just as a representative trajectory of a cell, we've marked out now where it hits this resting state. And what seems to happen is that the cell is periodically returning to this gait state 1, and the intervening transitions are a mix of these unbalanced, non-equilibrium-like transitions and more equilibrium-like balanced fluctuations. And it's possible perhaps that the cell is sort of pushing the cirral activity into particular kinds of regions of state space where it then fluctuates around before finally returning to this sort of resting state. And I'll also mention that these sort of returns to the resting state are consistent with the kinds of velocity fluctuations that people have seen in the overall movements of the cell. So what this picture of the gait is that it's a very stochastic gait. But nevertheless, there's still stereotypy, and there are these sort of cycles of activity that we see. So why might the cell walk in this weird kind of way? This is very speculative and hand-wavy sort of explanation, but it turns out in nature, these cells aren't just walking across cover slips. They're spending a lot of time in all sorts of complicated environments, and perhaps this kind of stochastic gait robustly allows them to migrate in complicated environments, such as this fungal hyphal network that and biofilm that the cell is crawling around on. And I also emphasize that you might think that, okay, this non-equilibrium character to the gait is not really surprising. The cell is walking obviously in a line, but even non-equilibrium systems don't necessarily have to obey this sort of broken detail balance, non-equilibrium type behavior at the sort of cell or mesoscale. And random walkers, this well-known sort of run and tumble motility that we see in E. coli, and there are similar kinds of movements that we see in flagellates as well, that can be mapped onto an equilibrium process. Some other work in the Marshall lab looking at sort of the fluctuations in shape of cells as they're hanging out on cover slips. So adherent cells, that can be described in an equilibrium model. And even though it's obviously an active process, it's consuming energy as the cell moves around. And I'd also emphasize that this kind of stochastic gait is different from something that we see in a lot of other organisms, including other ciliates, where we often see, if you have a lot of cilia all over the cell, this is a paramecium, you'll see metachronal synchrony. So the behavior of the individual cilia is much more determinate. So this kind of stochastic non-equilibrium gait represents a departure from some of the best characterized systems. And it is perhaps, though, a bit similar to other kinds of state transitions that people have examined in other kinds of systems. In this case, the gait transitions in a flagellate cell also show this characteristic broken detailed balance. And perhaps some of the hunting behaviors that people have looked at in this other ciliate called Lacrymaria also might exhibit these kinds of non-equilibrium transitions. Okay, so how is the cell actually controlling all of this? And we're just starting to get an idea for the mechanisms underlying this kind of serial control. And I'll briefly tell you a little bit about that now. And what we've first done is actually returned to this crazy idea of the neuromotor apparatus. And we've reconstructed, this is tubulin staining of Euplotes cell, reconstructed this fiber system. What we've found is that sort of contrary to one of the standing hypotheses in the field, the connections between these fibers, so these are microtubule-based fibers, there are two different kinds that we just morphologically classify. There are these thick fibers you can see in cyan, and these thinner fibers that are less linear than the thick fibers in magenta. We find that the cirri, the interfiber connections associated with different cirri, so these are the base of all these different cirri that I've labeled, these fiber systems are completely connected, and the connections between these fibers don't actually straightforwardly correspond to the behavioral modules that we saw, these dynamical modules that the cirri are grouped within based on shared mutual information. It's not the interfiber connections alone that explain that shared mutual information. So, one standing hypothesis in the field is, okay, if you connect two cirri by these fibers, these interfiber connections, maybe there's information straightforwardly shared there. That's not what we see. But what we do notice is that cirri that have similar kinds of cortical connections associated with their fiber do seem to tend to be share more mutual information. So, for example, F and G, these two cirri, share a lot of mutual information. They actually don't share any direct fiber-fiber contacts, but they do have these very similar structures in where their fibers intersect with the cell cortex. In contrast, we have fibers or cirri D and E, which have very different fiber structures, and even though the cirri are relatively close to one another, they share very little mutual information, and that's at least correlated in part with these differences in the fiber system. And so this isn't to say that this is a real mechanism, but it at least gives us some hypotheses to work with, and it's interesting to consider. Okay, so just to give you kind of a summary of what we've done and what I think might actually be general in its applicability to many other systems is we took this interesting behavior, we quantitatively characterized it, we performed dimensionality reduction to identify these dynamical states. We then identified patterns and state transitions within the system, and that led us to look for cellular structures associated with the kinds of modularity that we were seeing in the dynamical patterns. And I think that this might be a more generally useful approach to take to understand cellular behavior in different kinds of systems. And this is something we're very much interested in doing. So unfortunately, the coronavirus really slowed this progress substantially, but we're very interested in making comparisons among other kinds of walking ciliates, of which you might be surprised to find that there are many different kinds of walking ciliates. And it's interesting to consider how such behavior might have evolved and how it's possible that the cellular structure can change while maintaining walking behavior. And we'd also like to extend to systems that are at least appear much different from walking and ciliates. And the behavior of things like amoebae, for example, while the sort of coarse graining into a state space might be a bit less straightforward, and that's generally kind of a problem in applying this approach to many other systems. Amoebae nevertheless exhibit highly stereotyped kinds of behaviors, and it might be interesting to see what kinds of parallels we find between how Euplotes moves about in terms of the dynamics, and also perhaps principles of control between these much more disparate kinds of cellular systems. So with that, I would like to, I guess I moved through this a bit quicker than I anticipated, but I'd like to thank you all for your attention. And I'd also like to thank the Marshall Lab for being a really great place to do this kind of a bit out there work that I've really been enjoying. And also, of course, thanks to funding.
Thank you so much, Ben. That was amazing. I really like talks with basically a lot of pictures to me.
Sure.
You have those things scrolling across the screen. I have a couple of questions myself, but there's a lot of questions in chat, so maybe we can start with those.
Great.
If you could read the question before you answer it, that's going to be helpful to people that watch the recording.
Okay, let's see. Okay, so someone is asking if I could speculate when the random walk, I guess when the walking could be reconstituted in vitro. I have no idea. I expect that, and Kate mentioned this in some brief email correspondence, that motility is probably a very tough thing to reconstitute. And I would think that something like this walking behavior would be extremely difficult. But nevertheless, I think that there might be some interesting control principles that may be useful in sort of in vitro reconstitutions. But I would think that it may be a long time before something like this could be reconstituted. We'd have to learn a lot more about how the individual cirri, how those movements are actuated, and that presents a very interesting problem in its own right of how you get coordination of the cilia within one of these leg-like appendages as well. So I think a while, but it's an interesting kind of question. Oh, okay, so people are asking about sort of reconstituting this sort of thing in another system. I think that's a very fascinating question. And I don't have a background in this kind of synthetic biology. So I guess what I could say is maybe from a more purely biological perspective, it's interesting to me to think about how you would pattern a cell in order to get one of these cirri. And maybe there's something that we can learn about the differences in sort of cell patterning between ciliates that have this diffuse covering of cilia all over them versus these more discrete kinds of structures. And it would be interesting to think about changes in the genome that would be required for that to happen. Okay, let's see. Is there any protocol for going through these questions, or should I just?
I will usually, I think, go in order in which they were asked, but up to you. Sure.
Okay, so then someone is asking, can we use this model to train or regulate T cells to capture and kill cancer cells? I love that idea. I think that the sort of application for something like that is not so straightforward, but I think in terms of understanding something more about what cells need to actively regulate and actively sort of keep track of in order to properly execute a functional behavior, I think maybe there could be some general sort of shared principles there. But in terms of making that transition from something like a ciliate that's so different to something like a T cell, I think that the gap is quite wide. But I do think that there's at least an opportunity for some shared principles. Okay. Do I think that this model will generally apply to both cilia and flagella movement? So I think of cilia and flagella as more or less the same structure. And so I guess things that we learn about the movement of the cilia here in principle would apply to flagella. As far as I understand it, generally when people make the distinction between the two structures, flagella are usually longer and there's usually only sort of one of them. And I guess maybe this doesn't answer exactly this question, but I think something that's fascinating is the movement of these cirri. As I said, they're composed of many cilia. The kinds of movements that they execute are actually very similar to the kinds of sort of power stroke recovery strokes that we see in individual cilia and flagella. And so, I mean, that perhaps speaks to the generality of the constraints of low Reynolds number kinds of movements. And so I think there could be some interesting parallels to draw there. Although I would also emphasize that the mechanisms driving the movement of a cirrus versus a flagellum are at least sort of different. So instead of microtubules sliding around, now we have individual cilia that are sliding around in order to execute the sort of bending movements of a cirrus. Okay. What about the movement of simple multicellular organisms? I really like this question. I think, again, that there are interesting differences in the way that Euplotes is moving compared to what animals tend to do. As I said before, it's much more stochastic in the movement of the limbs. And that represents a very interesting sort of difference. But I'd like to think that there may be, again, some interesting sort of shared principles in gait coordination that we can extract. And it's hard to see exactly what those might be, but I'm certainly inspired in the kinds of experiments that I'm gearing up to do by what people have done to understand gait coordination in animals. And maybe a bit less horrifically, a lot of people do ablation experiments to study things like motor control and gait coordination in animals. With the cell, it makes me cringe a little bit less to think about doing those experiments, but Euplotes is animal-like enough that I do get a little twinge of guilt in thinking about cutting the cell or cutting its legs off. But I think those kinds of experiments could be very, very informative. And so while I think the models that one would apply to understand gait coordination could be quite different, the procedures to understand gait coordination could in fact be somewhat similar. Okay, what about movement of metastatic cancer cells? Again, this is pretty far outside of my depth to try to answer this question, but I would say that one possibility that I could see in relationships between the kinds of analyses that I've done and what we're seeing in cancer cells, or what one might see in cancer cells, is that perhaps breaking the system into these discrete states and looking at the state transitions, that might help in predicting future state transitions of the system. For example, you can imagine that if you see these unbalanced kinds of transitions in your analyses, that might give you insight into what the cell is actively controlling in terms of its behavior. And if you can identify sort of the mechanisms or cellular structures that are associated with that kind of control, that might give you some power to predict what the cell is going to do based on observing its current state. And it might also, give you the ability to stop it from doing something that you don't want it to do, if you can identify those kinds of control points. Okay. So this person is asking me about diagnostics like AIC or BIC did I use to evaluate the gait states? So I think this is about clustering. Okay. So, and then they're asking, are these 32 gait states clustering, are they universal for every cell or for an individual one? So I performed the clustering on sort of the aggregate of all of the data over a bunch of different cells. And it's interesting to think about individual to individual variability, and I haven't done much analyses there. As to the first part of the question, I'm not familiar with AIC or BIC, but I can say that for the cluster analysis, I looked at things like silhouette coefficients, and also did some cross-validation where I looked at cluster stability in the presence or I guess in the absence of randomly selected data points. So how stable the clusters are if I start taking away points. And hopefully that addressed some of what they were asking about. People are saying that they want to see a synthetic crawling cell. I would love to see that also. Any of you engineers out there, if you're interested in talking with me more, I'm happy to try to help as much as I can. Okay, so someone is asking about dimensionality reduction. So that I've done it's called non-negative matrix factorization. And it's a matrix factorization a bit like PCA, except instead of enforcing orthogonality of the components, it enforces that you have a sparse representation, and also that the components have only positive values. So part of the reason that I chose non-negative matrix factorization is that it makes the axes of my reduced state space very interpretable. Like I said, I can find these individual groups of cirri, and the activity of those cirri is associated with the basis vectors in the reduced state space. So, okay. Someone is asking about signaling pathways through which it moves. And And I have not looked at any of that at all, but I am certainly interested in perturbative experiments that would allow us to start untangling these kinds of signaling pathways or molecular mechanisms involved in gait coordination. And what I think we do have now is this platform where we have a really good description of the gait. And so it gives us this opportunity to introduce perturbations of various kinds. I can imagine RNAi experiments, drug treatments, ablation experiments, like I said. And even in the case where we somewhat subtly perturb what the cell is doing, since we have this good quantitative characterization, we might be able to better interpret what's happening in the cases of disrupted walking. And I think that's one strength of the kind of quantitative approach that we've chosen to take here, is that we can introduce a bunch of different kinds of perturbations, and we can really look in a bit more detail as to what aspects of the gait are being altered, even if we don't completely stop the cell's ability to walk. So the real answer is we don't know really anything about the molecular mechanisms, and we'd love to learn more. And that's absolutely one direction that we're going. Okay, so here's another person asking if peptide structures are causing motion in the organism. And then they're asking sort of a follow-up related question, was motility invented, or did it evolve several times in different organisms? So, I mean, certainly in very distantly related organisms, if they do anything like walking, it's completely independently evolved. I think it's not entirely clear in the group of ciliates that display this kind of walking behavior. It's not clear whether that was ancestral to them all or whether it may have independently evolved. I don't know the phylogeny well enough to give a super confident answer, but I believe that it seems as though walking may have evolved separately in different lineages. As far as what is causing the motion of the cell, I guess I can interpret this question somewhat broadly. One virtue of looking at a cell like Euplotes, it's a protist, is that it lives in a pond, and it's relatively easy to watch its sort of natural-like behavior. Granted, its natural environment is not walking across cover slips, but I can just set cells down on a cover slip and just watch them walk around, and they will just do this sort of without stimulation. But these cells also respond to a lot of different kinds of stimuli, and they respond often by changing the way that they're moving. So they have this great escape response that you might have noticed in some of the videos that I showed, where if the cell experiences a mechanical stimulus, for example, or dramatic change in pH or temperature in its environment, they'll escape, and those rear cirri sort of flick backwards in almost like a shrimp-like, I think of it as a shrimp-like movement, and the cell very rapidly moves backwards. And so that is a highly reproducible behavioral response that we can evoke using chemicals or physical stimuli. Also, the cells sense and respond to pheromones that are secreted by other individuals. And so they can recognize their own pheromone signal, but there are a bunch of different mating types, and there's all sorts of complexity associated with that, but they can recognize the pheromones of other cells. And that tends to change their walking behavior as well, where they will execute more turns. So they'll walk in less linear trajectories, and they will also start executing this escape response that I talked about more frequently. And so what that will tend to result in is that the cell stays in a much smaller area. So they can kind of sense and find one another and stick close by. So it's kind of interesting. People are noticing, they're making nice comments about Skeena the puppy. Skeena is my dog, actually. She's a great puppy. Okay, let's see. So this person is asking about any thoughts or comments about tracking and recording the vector fields. How about introducing, so to try to summarize this question, they're asking about Z-stack changes. So I presume this is the Z dimension and how the cell is moving in that dimension. And how would my analysis change in 3D versus 2D analysis? So again, I guess I would emphasize that this is another aspect in which the Euplotes behavior is nice for quantitative analysis. It's that this walking behavior does happen on surfaces. And so it is inherently a two-dimensional kind of behavior. And so I guess there are two ways that I could imagine introducing three-dimensional information. So one of them is that it would be amazing to have full three-dimensional information about what, for example, the tip of a cirrus is doing as the cell walks around. And that might allow me to derive a more fine-grained state space, where I could imagine, instead of just having this binary vector of, the cirrus moves or it doesn't move, I could say something about the shape that of the beat form of a cirrus movement. And so that might allow me to have a more fine-grained behavioral analysis. I think that this kind of information will become important if I start to look at things beyond this simplest walking trajectory. For example, these cells can turn, and these sort of escape responses, the cirri are doing all sorts of other interesting, complicated things. And that kind of three-dimensional information could be very important for understanding what's going on. Additionally, that sort of 3D information will be useful for understanding how an individual cirrus is controlled and how these beat forms are actually actuated. I guess the other question about three-dimensional information might be what the cell itself is doing in 3D. And these cells do swim. I didn't really address that at all. But I would love to get 3D information about, for example, the cell swimming around in solution, finding a surface and then landing. I think that's a very interesting kind of behavior. And that would require 3D information. And I could do a lot more different kinds of behavioral analyses if I had this sort of 3D information. But even if I were to do these kinds of analyses, I think absolutely the underlying information would change. But I think that sort of the framework that we've developed in terms of this dimensionality reduction, coarse graining these behavioral states, I think that could proceed in a somewhat similar fashion. Okay, next question. Someone is asking, are there perturbations to break the typical gait states that I observe? I think I kind of touched on that already in previous questions, and I haven't done any careful analysis of these, but yes, there are ways that I can perturb the walking of the cell. For now, I've mostly been focused on drug treatments. Things like lithium tends to mess with the walking of the cell, and that drug treatment has been used to paralyze flagella in other systems. But I'm working on drug treatments with more specific kinds of inhibitors. I mentioned that there is electrical activity in the cell associated with walking behavior. And so things like ion channel inhibitors become very attractive. I'm looking forward to seeing what those kinds of experiments show. Also, I mentioned these ablation experiments, I think will be very informative as well. Okay. Someone is now asking if I've noticed differences in cilia movement and cell behavior in the presence of gradients. I think I addressed that before with these interesting pheromone results. And I should also emphasize that, people have been studying the cell for over 100 years. People have made a lot of really interesting observations about the behavior of these cells. And as I said, there are these interesting pheromone results. People have even done these very difficult electrophysiological experiments to look at electrical activity in cells that are sensing versus not sensing pheromones. But again, there hasn't been this careful characterization of the dynamics of the cirri. And I think that I'm very interested in looking at more generally how the environment affects the behavior of the cell. So pheromones are one way to do this. Food, they tend to behave differently once they've actually physically encountered food. Again, there are all sorts of interesting physical stimuli that they respond to as well. So yes, they respond to a lot of different things. Someone is asking about how the Euplotes swim. And as far as I know, and I haven't been studying for Euplotes for so long, so I'm certainly not, a super expert in all of the amazing things that they do. But in asking around, it sounds like the Euplotes tends to swim using the membranelles. So I didn't talk about this structure at all, but the cells have this feeding apparatus. I guess you can't really see it in this image, but you can see the membranellar band of stentor here, these bands of cilia that draw in feeding currents. And so the cell, to feed Euplotes, actually has to sort of stand still, and it beats these membranelles, and that pulls in a feeding current, and it sort of filters food items out of that flow. When the cells swim, they seem to sort of tuck in their cirri, and they use the membranelles to swim. Although it's also possible that in some of the swimming maneuvers, the cirri become involved. For example, in the reorientation of the cell as it's going to land on the surface, perhaps they're involved there. But it seems as though a lot of the swimming, and they're actually quite good swimmers, a lot of the swimming is driven by membranellar bands. It turns out, somewhat interestingly to me, that other somewhat related ciliates that also have this walking behavior, 𝗂𝗶𝗶𝗃𝗃𝗃, for example, is one of these. They can't swim very well. And it's interesting to think about sort of maybe trade-offs in swimming ability and feeding ability, and also these walking behaviors as well. Okay, one last question now is, do changes in membrane potential correlate with particular gait states? Yes, it seems that they do, but not necessarily the gait states that I've identified here. Okay, so back up just a minute. The gait states that I've identified are particularly with respect to this linear walking behavior. I would love to be able to do imaging of cell potential or other recording of cell potential to see if as this question asks, if these kinds of gait states or gait state transitions are associated with any aspect of the membrane potential. And I think that's a good hypothesis for at least part of what's involved in controlling the transitions that we see. So the real answer is, I don't know. I would love to know. But in terms of higher order gait states, so transitions between linear walking behavior and these escape responses that I mentioned, and these sort of turning behaviors that the cell might execute, they can walk in little circles. Those higher order sort of transitions to completely different kinds of gaits, those transitions do seem to be driven by changes in the electrical activity of the cell. So I think that is a very interesting sort of direction of research, although the experiments are technically very challenging. And with that, I think I have made it through all of the questions.
Thank you so much. You basically gave a second talk on top of your first talk.
There are a lot of really good questions, so I'm happy to answer them.
That's awesome. All the questions that I had were already asked by other people in chat. So I just want to say thank you very much. Give a hug to Skina from me.
I will.
And thank you very much again for giving the talk.
All right. Thank you for inviting me. This was very fun.
And thanks, everyone, and have a great day. Bye.